I did not do
grades are
it.
still good in this class. Actually, I almost have B's in chemistry and calculus now.
That's awesome. Actually.
Just gotta get better on every exam.
Yeah,
After the first
I'm
one.
on like the precipice of dipping down into a B. I'm holding my A, and it's just been like slowly shrinking.
Yeah.
I gotta hold it. Gotta hunker down for the rest of the semester.
Gotta really lock in.
I'm the opposite. I messed up in both those classes, so I'm like, I gotta work really hard to get it back.
I think I'm gonna have straight B's this semester, but that's fine, I guess.
Okay,
Yeah.
all right.
I'm good with an A and a couple B's.
I'm gonna give out your lecture exam first, and then we'll go over that,
Look at this
answer any
little
questions.
thing I got.
If you have any changes to
I
make,
really
do
like
that.
your sweater too.
And then we'll go over your lab
Where'd you
exam
get it?
next. I don't want to put them
just
both at the
ordered it
same time,
online
though, so I want to make sure I get them back. I have entered the lecture exam, but I will.
I'm gonna learn
will
that I
check to
need
make
stuff
sure
like
we're
this.
returned to them. I have not yet entered your lab exam to the Canvas yet, so if I don't get it back, please do. Okay? So make sure I get that back. The only thing I already entered in the Halloween costume contest. Very good. We had 19 costumes, so 2 people tied for 19 points, and then it was down from there. No one got less than 10 points. So it's, I think the thing is the judges graded like creativity and effort, and even though the kind of store-bought costumes were cool and everything, you know, the idea was to have something that was really kind of clever and creative. So Praying Mantis got first place, yay! And then it was the Dissecting Frog that got title. And then everyone else is great. All the jellyfish and insects were just very impressive. Okay, and this is the record. I've never had that many students do dress up, so it's great. Good job for all of you. I've never given out that many credit points, so oh well. That's the way it goes. That's what I promised I'd deliver. Okay, so this is your lecture exam. It's out of 100 points. Someone got 100. So either someone has a PhD in biology and didn't tell me and just hanging out here for no reason, or they studied their rear end off. So that's why they're Telson off. So they're Telson off. Okay, Rebecca Johnson. Yeah, cool. And JD. In no particular order. So out of 100, so right now I just have it as 100. I haven't curved this one because I said I would use the top.
The same score in both.
May.
I mean, 87 throughout the class.
Elijah.
Oh,
Yeah.
I'm stupid.
Sage. No?
The next one next.
Louise. Caleb. Okay. Caleb's here, that's all that matters. Kendra. Okay, here you go.
I like the blue again.
Brittany.
I like these pens so much. I'm so glad I got
John Lotfi. No.
lecture
Good job.
exam.
Daniela.
Don't say my name and I wasn't paying attention. I hate when I do this.
Okay, Clark. No?
No Clark.
Lene. Okay, great. Canyon. Acacia. Mia. The other John. Here you go.
I'm actually surprised on some of these that I did like, okay.
Shanna. Joelle.
I
Brooke.
love myself so much. I get those kids right.
Ariana.
Yeah.
No Ariana today?
Yeah.
Caitlin. Easter. Yep.
I also got -6, just in a different way.
Peyton. No Peyton.
It is what it is.
Chris Nichols.
Where did I put these?
Carly.
I knew this part would be bad. I made up for it with
Hey.
everything else.
You, Riley.
Take what you
Benny. Joshua.
I don't know.
Riley, boys player.
It's actually fair.
No? May.
Lost it was like what? We lost it.
Elijah Davis. No? Sage. Okay. All right. Did everyone get a test back? You took one? Did I get everyone a test back? All right. So what I want to do is I want to go over these and let you know what I was looking for. These things took me forever to grade. And sorry about that. I try to get it back as soon as I could, but I had so many things to grade and things to do. But anyway, finally got them. But because it was so written, it was like just took me forever. So anyway, I struggled. So anyway, let me tell you what I was looking for. And if you believe you have what I said I was looking for, then I will take a look at it again. Chris, what do we got? Peyton? Or no, who's it? Chris, that's right, Chris. Chris here. Elijah, I'm sorry. Elijah Davis. Okay.
When the exams makes me feel happy.
Okay, so if you think you have what I was looking for, make a note, and then we'll have a moment afterwards for you to kind of argue that you had what I was looking for. And if the person next to you wrote the same thing as you did, and they got it right and you got it wrong, bring me their test. I'll mark it wrong in there too. Okay, so we have the mosquito in salt water. If the mosquito is 1% saline and the salt water is 3.5% saline, remember, hypertonic means more solutes, right? Hypotonic means less solutes. So the salt water is hypertonic and the cells are hypotonic. And that means the water goes out of the mosquito. So the arrow should be going away from the mosquito to the water. And the opposite in the fresh water, the fresh water has less solutes, so it's hypotonic and the cells are hypertonic, and the water goes into the mosquito. Okay? Water loss to the atmosphere is called desiccation, Desiccation. I had that on the study guide. That's not a question. So I felt it was fair enough to ask that. Okay. Then question 2. It says, what are the 3 potential byproducts of protein metabolism? What are the advantages and disadvantages of each? Okay, so you didn't have to have them in order, but it made it a lot easier if you had them in order. So if you put ammonia on the left or right, but if you put ammonia on one side, and then in the middle you put urea, and on the other side you put uric acid, that made your life easy because you could just kind of write more simple stuff, right? Because urea is kind of in the middle ground, so you didn't really need to specify much about urea other than who makes it. So for ammonia, it has high toxicity. Uric acid has low toxicity. Urea is in between. For ammonia, it takes no energy to make. For uric acid, it takes a lot of energy to make. Ammonia is very soluble in water, so that's a benefit, meaning you can get rid of it easy, but also means you need a lot of water to get rid of it. Uric acid is not soluble in water, okay? And so I think those are the 3 main things that I wanted to see was the toxicity, the energy to make, and the solubility of the water need to get rid of it. And for ammonia, it's small aquatic organisms. For urea, it'd be like mammals. And for uric acid, it'd be like insects and reptiles. And there's a wide variety of answers there, but there you go. Okay, and then question 3, compare and contrast. Some people, I don't know if you just didn't read the question or if you misunderstood the question, but I'm asking about the water balance and nitrogenous waste excretion. And some people gave me like an elaborate detail of how they eat. And I'm like, okay, so I had a hard time, I tried to see what I could do here. But anyway, for the water balance nitrogenous waste, it was pretty simple. For the hydra, for water balance in the marine environment, they're osmoconformers. But in the freshwater environment, they use that gastrovascular cavity gut pump, right? They use their gastrovascular cavity to pump water out, and ammonia leaks out of their cells. They're ammonia excretors. So I think the main thing I'm looking for is they don't have any specialized organs for getting rid of waste. They get rid of ammonia. And in freshwater, because they have to be osmoregulators, they use this gastrovascular cavity pump to do that, a behavioral thing. In the planaria, they have flame cells in the freshwater environment that they use to pump water out. But I was emphasizing in class that they also just leak ammonia out of their cells. They don't use urea or uric acid. They just get rid of ammonia by diffusion. I know the book says that the protonephridia get rid of ammonia too, but ammonia just leaks out of the cells. So their flame bulbs are for osmoregulation, but not for nitrogenous waste necessarily. And then the earthworm, I think the big key feature here is in the earthworm you have an organ, the nephridia, and I was hoping you'd write nephridia, that both regulates water balance and gets rid of nitrogenous waste like urea. So I guess I'm kind of looking for a progression. The hydra, where they don't really have any specific organ for waste or water balance. The flatworm, where they have an organ for waste, for water balance but not waste. And then the annelid worm, where they have a combined organ that does water balance and waste. It's kind of the idea, right? I was trying to see what you knew and help you out. It's worth 6 points, so take a look there. Okay, and then here we have, again, I say compare and contrast the digestive system. We're on page 2. And some people gave me elaborate details of other things besides the digestive system. So again, I don't know if it was a disconnect or if you were nervous or if you didn't read the question or what's going on. But for the porifera, the digestive system is, they essentially are filter feeders, and they need to feed on food that's smaller than their individual choanocyte cells. So they have to feed on microscopic food particles that are smaller than their feeding cells. For the jellyfish, they have a gastrovascular cavity, which means that they can ingest food, break it down, and they can digest things that are larger, like a cavity or a pocket. And again, the progression here, the nematoda has a gut tube or a gastrointestinal tract. So I kind of wanted to see the Nigeria having gastrovascular cavity, or you could say gut pocket, and that the nematode has a gastrointestinal tract, or you could say gut tube. That would be fine. But again, we're looking at a progression here that kind of summarizes what we've seen in these animals, the complexity of digestion. Gotcha? So there you go. Again, that's worth 2 points each idea. And the only reason I say draw a diagram is because I think sometimes the diagram helps you not to write so many words. And so it makes it a little easier. I don't expect everyone to be like a total artist, an amazing illustrator or whatever. But sometimes if people just drew me a diagram but didn't write anything, it was hard for me to know what, I had to kind of interpret that. I tried to give you the benefit of the doubt. If you drew a nice jellyfish with a gut pocket, I'm like, all right, okay, that's fine. Okay, question 5. Most animal species require a large supply of oxygen for respiration. Why is oxygen so important for animals? And what happens to animal cells when oxygen supply is limited? Okay, so there was like a really specific answer here that would have been great. I mean, some people gave me this. But basically, with oxygen, animal cells can convert about 32 or so % of the energy found in their food to cell energy. So some people told me the percent, it's like 34% conversion to ATP. Some people gave me the number of ATP, 32 ATP. Something that resembled that idea is that they can convert about 1/3 of the energy of their food into cell energy with oxygen. And they can only convert less than 2% of that energy from food to cell energy without oxygen. That's kind of the best answer. You didn't have to tell me number of ATP, just kind of like it's like a big concept. 1/3 versus 2% is a pretty big difference. And because animal cells, because muscles and nerve cells require so much ATP, most animals can't live without oxygen supply. Plenty available. So, some people kind of worked on their expertise with tautology. Like their answer would be something like, animals need oxygen because they need to breathe. It's like, all right, well, yeah, you can never be wrong with tautology, right? So anyway, but I needed something really specific, which was something about that they get 15 times more cell energy with oxygen versus without. Would have been also fine too. What kind of animals can live anaerobic without oxygen? I mentioned gut nematodes was a common one, but some people mentioned the little cnidaria that lives in salmon mussel. That's fine. But gut nematodes was the easy one. Okay, 6. What are the basic advantages and disadvantages of each type of animal food below? This was really simple, and I was looking for just major key things. The advantages to carbohydrates is it's quick, ready energy. The sugars, especially, provide for fast cell energy. It's kind of what the basis of our metabolism description is. If you compare it to fats, and here's what I kind of wanted to see. Fats yield about twice as much energy per mass, or some people wrote more specifically that they produce 20% more ATP at 2/3 the mass. Just some indication that fats yield a significant amount more energy per mass unit versus carbohydrates. So I don't really care about fats being healthy or unhealthy. It's just they're more efficient because you only have to eat half as many fats per mass compared to carbohydrates. You get the same amount of calories, right? So fats and oils are more efficient by twofold. And then proteins are about the same energy as carbohydrates, but the thing I needed to see was with a protein metabolism, you make ammonia. So the amino breaks off, and ammonia is a toxic waste. So indicating that proteins produce a toxic waste, namely ammonia, is the main thing I'm looking for there. So I guess the key minimum would be carbohydrates is fast, easy energy. Lipids have a little bit more than 2 times the energy, and proteins have the same amount of energy as carbohydrates, but have a toxic byproduct. Got it? Okay, so there's a lot of variations in there. You know, some people gave me nutritional rundowns. I think someone tried to sell me the Atkins diet here. So anyway, that's the way it goes. Okay, any questions there? Question 7. This is a compare and contrast. Some people did really well on this, super well. So I think I probably gave you smiley faces or whatever if you did a good job on this. So ideally what you do in a compare and contrast is you kind of briefly describe how the terms are similar. That's the comparison. And then the contrast would be how are they different, right? It's kind of the idea. So homologous versus analogous traits. Basically, we're talking about adaptations here. And in homologous traits, it's adaptations that derive from common ancestry, even if they're used for a different function. Analogous traits are traits that are derived from a similar function, but a different ancestry. So analogous traits would be like, say, for example, insect wing and a bat wing. They're wings for function, but they come from a different ancestral lineage. It's kind of the simplest answer. Vestigial traits versus exaptation. How they compare is basically change in adaptations. In the vestigial trait, the adaptation is reduced or disappears, and in exaptation, the adaptation is converted to a new use or a new function. It's kind of the idea. Okay, osmoconformers versus osmoregulators. This was a lot easier than I think some people thought. Osmoconformers maintain their internal cell osmolarity or solute concentration to match the external osmolarity or solute concentration. So essentially they keep their inside equivalent to the outside's osmolarity, so they don't have to worry about doing water balance. Whereas osmoregulators maintain a constant internal osmolarity or solute concentration, even if the external environment is changing or is different. Some people just wrote osmoconformers marine, osmoregulators freshwater. I mean, that's true, but it's not really defining what the term means. So you got partial credit for that. But yes, it's true that, you know, freshwater organisms have to be osmoregulators. But I kind of needed to see what that means, right? It's more than just those two words. Okay, tonicity versus osmolarity. Tonicity is basically the concentration of solutes in a water solution or a cell. Whether it's saline or sugars. And osmolarity is the potential for water to cross a membrane. So if you have a osmolarity situation, you basically have the potential for water to be drawn across the membrane. So I gave the example in class that like a 0.9% saline solution, it has equivalent osmolarity as a 5% glucose solution. They both have the same potential to draw water across the membrane, right? That was kind of the idea, is tonicity is the solute concentration, osmolarity is the movement of water across the membrane. Okay, acoelome, pseudocoelome, and a coelome. The acoelome is an organism that basically has just a gut pocket, but the mesoderm has no other pockets or any other cavities associated with it. And the example, you didn't have to give an example, but it'd be great if you did. It was the flatworms. The pseudocoelome was like the nematodes. Again, you didn't have to give an example. But a pseudocoelome is a cavity around the gut, but the gut is not entirely lined with the mesoderm. And then a true coelome is like in the annelids, where there's mesoderm lining the gut and the outside of the body cavity. So it's a cavity entirely lined with mesoderm. That's the best definition. Some people gave me like perfect textbook definitions. Others I had to kind of extract that you understood what it meant. And so here's the problem again, like saying acoelome, just saying no cavity is not, I mean, it's just not quite enough. And then pseudocoelome, just saying fake cavity, I mean, you're just basically telling me what the word means, you're not really saying what it is. And then the true coelome is kind of like the basic reference point. All right, protonephridia versus pseudonephridia versus nephridia. The protonephridia are the flame bulbs found in flatworms. They're basically water pumps that use cilia. The pseudonephridia are the excretory tubes found in nematodes, and they don't have cilia. And the nephridia are the ciliated tubes found in mollusks and segmented worms, and that's kind of the main definition. It's a ciliated tube that filters the coelom of water and nitrogenous waste. So I was hoping that cilia would be referenced in there, and I probably wrote cilia if it wasn't. Okay, gastrovascular cavity. This is a gut pocket that has one opening, and a gastrointestinal tract is a gut tube that has a mouth and a separate anus. So it's kind of like really a simple answer. Gastrovascular cavity is a gut pocket with just a mouth opening. The gastrointestinal tract is a gut tube with two openings, one in and one out. And then hemoglobin versus hemocyanin. Hemoglobin is an iron-based protein that carries oxygen and turns red when oxidized. Hemocyanin is a copper-based protein that turns blue when it's oxidized. That's kind of like the bare minimum. Hemoglobin uses iron and it turns red. Hemocyanin uses copper and turns blue. Some people gave me even better definitions, like hemoglobin uses one iron to carry an oxygen, whereas hemocyanin uses two coppers to carry an oxygen. And some went beyond that, like organisms with a closed circulatory system have hemoglobin, and organisms with an open circulatory system have hemocyanin. But as long as you just gave me the main difference, which is the metal and kind of how they operate. That's good. Okay? All right. So, the stratigraphy, anyway, on this one, it was kind of a challenge. I was surprised that people missed, I thought there were pretty obvious, easy answers, but some people kind of missed the main points. So for the relative age of the rocks, all you really needed to say was A, because it's near the surface, is younger than F, which is deeper. Or if you indicated anything that said that the shallower rocks are younger than the deeper rocks, that was what the relative age was. For 8B, what two general types of rocks are listed? And that's sedimentary and igneous. So sedimentary and igneous rocks are the two general types. Okay, which layers are most likely to contain fossils? It should be the sedimentary rocks, and I say specifically, it's mudstone and shale are the two main ones. Some people added clay in there, and that's all right. But as long as you had the A and C, mudstone and shale, that was kind of the main right answer. And then this one, you know, I think it's obvious question, but very few people actually gave me the specific answer. And so potassium-argon test reveals two dates, 120 million years and 240 million years. To which specific layers can these apply? Well, remember, potassium-argon only works with volcanic rock. So that would be ash layer and pumice and lava are the only two volcanic rocks there. And so B would be the 120 million year because it's shallower. And D would be the 240 million year because it's deeper. That was what I was hoping people would connect. But like very few people made that connection. So a lot of people put B and D, which maybe is just a coincidence, so I accepted that as being the sequence. But trying to figure out how to get people to specifically say which one's which. Anyway. And then 8E, carbon-14 would be useful in this example. Or sorry, would carbon-14 be useful? And I mentioned in class that no, carbon-14 is really not useful in paleontology because it doesn't go old enough. Anything older than 60,000 years is really not useful. And nothing in here indicates that anything is less than 60,000 years. I mean, you could say, considering that layer B is 120 million years old, if layer A is less than 60,000 years, you could use it, but no one said that, right? So the main thing I'm looking for is probably not or no, because carbon-14 is too little a time, 60,000 years, to be useful. Got it? Okay. So, question 9. Compare and contrast the respiratory adaptations among the following organisms. Again, so, you know, we had water balance, nitrogenous waste, we had digestion, and now we have respiratory system comparisons. So, for the flatworms, they're basically using simple diffusion of oxygen and CO2, but I mentioned they flatten their bodies to enhance the transfer of oxygen into the tissues. So they do a behavioral flattening to enhance respiration in those. So hopefully that was said. In the annelid worms, they do diffusion across their tissues, but they have a closed circulatory system with hemoglobin to help carry oxygen and CO2, to move those oxygen and CO2 around. And then the main thing I'm looking for here is in the mollusks, they have a circulatory system also with hemocyanin, but they usually have an external gill, lymphanidia. It's a respiratory structure. So again, I'm looking for this progression of no respiratory structure, but behavioral change, respiratory adaptations internally, like with the circulatory system, and then respiratory adaptations externally with like a gill. Just looking at a progression there. Okay, and then what adaptations enabled mollusks to colonize land? And that would have been things like shells, slime to prevent desiccation. And the main thing I'm looking for is they've reduced their respiratory surface area. They've converted their mantle cavity into a lung. That was kind of the best answer. Or they've lost their ctenidia. Was a good answer also. Okay? All right. Page 5, the bear question. So this one, you know, I was trying to extract from you some key points here. So this is a fun question because it's sort of addressing the biological species concept and the genetic species concept. So it says there's 3 bear species in North America. Brown bears can hybridize with polar bears. So it's indicating that brown bears and polar bears can interbreed in the wild and produce offspring. And then it says, what does the genetic data on the graph suggest about the relationship among these bear species? So I was hoping that somewhere in there you would say, the general rule of thumb is, if there's less than a 2% difference, they're likely the same species. And if there's greater than a 2% difference, they could be separate species, you know, if you're looking at the human chimpanzee model. And if you look at this little x-axis, I was kind of hoping, because I didn't change this, I was kind of hoping that most people recognize that 0.02 is 2%, right? 0.02 is the same as saying 2%. I think some people miss that. Even though I wrote, this is important, right? You know, had an arrow pointing right to it, you know. So anyway, so in that case, you can see that the polar bears and the 1 brown bear 4 population individuals are all less than 2% difference. So they're probably all the same species. That's why they interbreed, and that's why they're less than 2%. That the rest of the brown bears, if you look at where they connect to the polar bears and brown bear 4, is about a little less than 3% difference. So what I was hoping people would say was that 2% is a rule of thumb, and it could be as much as 3 to 5%, which is what that paper said. The paper said for rodents, it's 5% should be always better. Just in the abstract, you didn't have to read the whole paper. Just the abstract said 5% is probably a better mammal indicator for percent similarity or differences in order to be the same species. You know, I didn't really need you to say anything about the black bears because it's clear the black bears are not interbreeding with the brown bears and polar bears, and it's saying here that black bears are about 8% difference, right? But that was kind of like, you know, this is a real example, this is real. And so, you know, I mean, there is arguments to say that polar bears are just a specialized type of brown bear, you know, that live in the snow. And that this is the genetic data to kind of demonstrate that. But what I was hoping really is that you just said that the 2% is kind of like our rule of thumb or our kind of threshold for what we consider to be for sure the same species versus maybe could be different species, right? Cool. I mean, it's only with 3 points, but I kind of need to see that 2% indicator. Okay, compare and contrast an open versus closed circulatory system. For an open circulatory system, you have a mixed hemolymph, it's low pressure, and the pericardium is basically just kind of sloshing the hemolymph around inside the hemocoel. And that occurs in mollusks and in arthropods. The closed circulatory system has a separate blood fluid versus coelomic fluid, and it's high pressure, and the heart is usually the largest cavity, and that occurs in annelids and vertebrates. And which is better? It depends. If you're small and you don't move much, an open system is better because it takes less energy. If you're larger or have a lot of activity, a closed system is better because it's more efficient for respiration transfer, but it depends on the kind of organism. So as long as I had the basic descriptions, it was fun. Some people gave me really good descriptions. Some people I had to kind of really read into it, see what you were talking about. Okay, the cephalopods, the squids. I had mentioned this is controversial. So in my opinion, or how I described it, cephalopods have an open circulatory system because they have a mixed hemolymph, and their systemic heart basically circulates the blood fluid in a hemocoel at low pressure. But in the ctenidia are high pressure because they're pressurized by their own hearts, the branchial hearts. So they have a modified open circulatory system that partially resembles a closed circulatory system because of high pressure. I thought this was fun because most textbooks say that squids have a closed circulatory system, but if you think about it, their ancestor doesn't. All the other mollusks have an open circulatory system. So really they have an open system that has been modified to function like a closed system. It's kind of the best answer there, right? Okay, cool. All right, and then the last page, let's see here if I can find one that has right answers here. All right, let's just go ahead and do it together. If you probably have it based on what I've written there. Okay, look, it's easiest if you go backwards. If you start from the oldest to the more recent, I think it's far easier to do it that way than to try to work from current backwards, right? So current to the past. Better to go from past to current. So let's start from the bottom. The Hadean is F, right? No evidence of life present. There's only one way I could ask that, right? I think everyone got that one right. The Archaean is A. The only bacteria-like colonies are the only fossils present. A. And then the Precambrian is, what's that, N? Amoeba-like eukaryotic organisms appear and the first sponge fossils appear, because sponges probably should be protists anyway, right? So N is the Precambrian. Then we get into the Cambrian, and that would be, where is it? B, right? Segmented worms and arthropods suddenly appear in the fossil record, is in the Cambrian. And then the Ordovician is J. Early plants colonized land and the oldest fish fossils occur, right? The Silurian is, which one's a Silurian? Isn't it the first arthropods colonized land?
It's D.
G? D. Chelicerates and myriapods first colonized land, the first arthropods, right? D is the Silurian. And then Devonian is, where are we at here? Which one is it? Devonian is E, right? Wingless hexapods and the first amphibians appear on land. E. Carboniferous is the first terrestrial snails and the dragonflies take to the skies, is the Carboniferous. And the Permian is trilobites and greater than 90% of all marine species suddenly go extinct here, right? Okay, then the Triassic, M, the first turtles appear following a mass marine extinction. That was a big hint there. Jurassic is the feathered birds evolved to fly starting in this period, is Jurassic. And then Cretaceous is an asteroid collision with Earth likely caused Tyrannosaurus rex to go extinct here. 1 year on this, I was mean 1 year, and the answer was most dinosaurs in the movie Jurassic Park actually occurred in this period. They're all Cretaceous dinosaurs. I thought it was fun. Okay, so the Tertiary is bats radiated as the only flying mammal type. The Tertiary is the mammal radiation. And then the Quaternary is humans migrate across the Bering Land Bridge from Asia to North America. So anyway, if you just switch to, I realize if you get one wrong, you get another one automatically wrong, right? So if you just switch to, I just put a -1 next to the 2, and I put my arrows showing that you switched them. If you had a cascading switch where it's like you got one off and then it caused you to get like 4 of them off, like if you missed 4 or 5, I probably gave you 1 or 2 credit. If you missed 5, I probably said -3 or 4, depending on how bad it was. okay? So I tried to give you a little bit of benefit of the doubt on that one. But I thought these were pretty straightforward. About 4 or 5 people got this question, so it was totally doable.
It's because the
Okay, so that is our lecture exam. What I want to do is I want to get this back before I give you out your lab exam. So let's take a quick break, and if you want to just give me your test, hand it to me. If you want to ask a question, go ahead and line up over here on the side, and I'll take a look at it and give you
I
a
just
second.
want to know what my